Good afternoon, everybody. Is this on? Can you hear me? Yes, good. Okay. For those of you who don't know me, I'm Ruth Karen. I wanted to welcome you to the third annual Vaccine Day here at the School of Public Health. Uh, this Vaccine Day, as the ones before, is sponsored by the Johns Hopkins Vaccine Initiative. And for those of you who don't know what the JHVI is, it's a program of our school. It was launched in 2008 to promote collaborative and interdisciplinary vaccine research, education, and implementation efforts to improve health worldwide. And JHVI now counts among its members 30 faculty from 5 departments and from multiple centers and institutes within the school. I know that all of you are here to hear our guest, Dr. Stanley Plotkin, and so I'm not going to take away too much of his time, but I just wanted to tell you a few things about what JHVI has been up to over the past year. Just highlight a couple of things: our newsletter, our new human vaccine research database, and our VIEW internships. So we launched this year, this is really the work of Amber Cox, but we launched a quarterly newsletter. This is actually the summer edition. The fall edition has just come out and is accessible on our website. There's a lot of information about work being done at the school having to do with vaccines. We would welcome contributions. You can contact Amber, and she'd be happy to put information that you have into our next winter edition. The other thing I wanted to mention is, so I talked to you a little bit about this last year, that we were getting a database up and going. And the impetus for this was really, we recognized that there was no place in the school that we could readily get information about clinical vaccine research that went on at the school. So we went to the effort to assemble a searchable database, and it's now up and running. It was launched in September. It has data from the past 10 years and will be updated annually. There are 28 faculty whose projects are in the database, over 200 projects. This is a sample screen. This year, at our poster session, we'll have a laptop set up with the database accessible, so you can go into it and search for yourselves. You can certainly search. You can search by investigator. You can search by sponsor. You can search by pathogen. And we think it'll be a really useful tool to foster collaboration in the school and, in fact, across the medical community. I wanted to say something about a program that we're really proud of that was launched this year, which is the VIEW internship program. We have 5 students from our school at WHO this year doing research. They're shown with the stars. They're next to Dr. Chan. They are our stars. The VIEW program stands for Vaccine Internship Experience at WHO. These are 12 to 24-week funded internships in Geneva for our students with mentors in the Department of Immunizations, Vaccines, and Biologicals. We give preference in this program to students who've taken vaccine courses here at our school. The first internships began this past September, and I can tell you that this has been a remarkable success. We have heard from our students that this is a life-changing experience for them, that it's opened their eyes to new career possibilities. We've heard from the mentors that they are going to be very sorry to see these students go. Thomas Cherian, my colleague at WHO, who is spearheading the program there, asked for projects for next year, and within hours he got responses from mentors that said, I want to send mine in right away because I don't want to miss out on getting students for next year. So this has been, I think, incredibly successful, and we're very proud to be able to offer this program again next year. We anticipate that we'll have positions for 3 or 4 scholars. Applications are going to be due in early February. We'll post information online probably toward the end of December, and we'll announce awards in March, and you'll be able to get more information about this by visiting our website. So I'm not going to delay things further. I just would like to acknowledge a few people. First, I would very much like to acknowledge Dean Clagg, who's been incredibly supportive of the Johns Hopkins Vaccine Initiative, and so, of course, by extension of Vaccine Day. I'd like to thank BD, who has sponsored this event. Michael Garrison from BD is here in the room, and I hope that some of you will have a chance to talk to him later today. I'd also like to thank Amber Cox, without whom this absolutely would not have been possible. Many of you have met Amber. She's the program manager for the JHVI, and she made everything happen and handled things beautifully. I'd like to acknowledge the Office of External Affairs, who worked very closely with us, particularly Monique and Jeff and Donna. And of course, and there's a very important omission here, I'd like to acknowledge our steering committee, Neil Halsey, Orrin Levine, Bill Moss, and of course, Kate O'Brien, who was on my last slide, but somehow got left off this one, the steering committee, who really helped us all put this together. And I'd now like to ask Dean Clagg to introduce our guest speaker.
Thank you, Ruth. And yeah, I want to congratulate you on the success of the vaccine initiative at the Institute, because I can remember it was a short time ago when you were in my office and telling me about this vision, and you've made so much happen in such a short time. It's great. And today's a great example, because after last year, I didn't think you could top it. You know, we had such a great speaker, and it was, you know, exciting and scintillating and fun. But you have, and it's my happy job to introduce Dr. Stanley Plotkin. I had the pleasure of having dinner with him last night. And the last time I saw him was in 1975, when I was a second-year medical student and doing a pediatric rotation, and we rotated with him one day. And usually I kept my mouth shut when I was a medical student, especially second year training. But we were going into the room of a patient, a child with chicken pox, and it was before, of course, the vaccine, and I had never had chicken pox. And so I said, well, you know, I got a quick consult from you. Should I go in? And he said, what's going to happen? So go ahead. So, you know, I was, you know, because as you know, as a medical student, you always think the worst. But at that time, I recognized what a sense of humor he had, and he was incredibly respected by everybody at Penn. He's devoted his professional life to vaccines, and you know, is probably the most well-known vaccinologist in the world. He's worked at, and in this brochure, there's a lot about his career and the awards. So I can't do your CV justice, because it's just you've done so much. But just to say, he's had a great impact at the federal agency level, working in CDC, in academia, and in industry. And he is on innumerable advisory boards related to vaccine and vaccine policy here and around the world, a reflection of his wisdom and his judgment and the respect people have for him. He's always been a strong advocate for using the best science to make decisions, has developed, as it says in the brochure, the rubella vaccine that's now used around the world, and is co-developer of the pentavalent rotavirus vaccine. He's been elected to the IOM here and to the French Academy of Medicine. He's considered the founding father of the Pediatric Infectious Disease Society. So there's lots of awards. I'm just going to read a few, but he has received the French Legion of Honor Medal, the Sabin Gold Medal, the Fleming Bristol Award of the Infectious Disease Society of America, the Medal of the Foundation Miro, the Maxwell Finland Award of the National Foundation for Infectious Diseases, and the Hillman Award of the American Society for Microbiology. He's written over 700 articles, and he is the editor of the first textbook on vaccines, which is now in its 5th edition. He co-founded the first journal devoted exclusively to vaccines. When he was 74, and I guess was bored, he decided to get a private pilot license, so he flies. He's an avid flyer. I'm a frequent flyer, but he's actually a real flyer. And he is, I guess most importantly, he is an emeritus professor at the University of Pennsylvania and an adjunct professor here at our school. We could talk a lot about him, but I know from my dinner last night that he knows a lot about wine and fine food. So, Stan, we're very pleased that you're here with us today.
Well, thank you, Dean Clagg, for that very generous introduction. I am very conscious of the honor of having been invited to give this talk. Do you hear me? And very conscious of the distinguished audience at this distinguished institution. So let me see if I can get these slides moving. Nope. Yep. We need to get back to the... This is the advance.
Yeah, no, but we just need to actually get out of.
Oh, wait.
Yeah.
Well, there it is.
Here,
Yeah.
let's see. Let's just see. There we go.
Okay.
Yeah, and let's just
Here.
show it in
Here,
this one.
right there.
Here
Okay.
we are.
Right. Okay. So this title is a rather conceited title. And the excuse for that, my excuse for that, is that Ruth Karen insisted that I give this type of talk. So if you think it is conceited, it's not my fault. It's hers. But essentially, I'm going to review a number of things that have happened in my life. And my excuse is that all wisdom, as Isla said, begins with memory. Of course, as one of your Baltimoreans said, the older I grow, the more I distrust the familiar doctrine that age brings wisdom. So, but I'll let you make that decision. At any rate, the first decade of my own life was featured by 3 vaccine-preventable diseases from which, in fact, I almost died. One was a pertussis, a severe pertussis. The second was pneumococcal pneumonia that put me into an oxygen tent for a while. The third was severe influenza, which actually did put me into coma, and I had Bell's palsy when I recovered. Now, in 1939, during that decade, the total advice of the Red Book with respect to vaccination was that every child be vaccinated against smallpox, immunized against diphtheria, against typhoid fever if necessary, and injected with rabies vaccine if the need arises. So in my lifetime, there has been a revolution in vaccines, and starting from very, very meager basis. So what have I learned during a lifetime? I'm going to give you 10 lessons that I've extracted from my own experience. The first is take advantage of every opportunity. Now, when I finished medical school and then went to the Epidemic Intelligence Service of CDC, at the end of my training, there was a list of assignments, and I insisted on going to the Wistar Institute Anthrax Investigations Unit. The Wistar Institute is a research institute on the grounds of the University of Pennsylvania in Philadelphia. I'll tell you in a few moments why I insisted. But anyway, I arrived at the Anthrax Investigations Unit, and soon after I arrived, knowing nothing about anthrax, having little interest in anthrax, there was a call from a pathologist in New Hampshire who said he thought he had a case of inhalation anthrax based on his autopsy findings. So I went up to New Hampshire and investigated the situation, and it turned out that there were 5 cases of inhalation anthrax in mill workers in New Hampshire. And as we learned, as we investigated and retrospectively realized that they had all had contact with a particular lot of goat hair coming from the Indo-Pakistan subcontinent, which was grossly contaminated with anthrax, the goats presumably having died of anthrax. Well, this graph shows the clinical course of the 5 patients, typical of inhalation. Anthrax, the prodromal period, and then the sudden septic collapse, toxemic collapse. 4 of the patients died. This paper, the paper we published in the late 50s, was actually republished after the bioterrorist incident in 2001, some 45 years later. So I think that may be a sort of a record. At any rate, at the same time, CDC was studying the protective antigen anthrax vaccine that had been developed at Fort Detrick. This is based on a protein produced by the anthrax bacillus, which forms a heptameric pore on the surface of cells of the host, through which the lethal factor and edema factor toxins pass and, of course, kill the cells and eventually kill the host. So this vaccine, consisting of that antigen, was being tried in mill workers. And the end of the day, when we put together the results, the vaccine proved to be highly protective against cutaneous anthrax and probably against inhalation anthrax, although the number of cases was small. This vaccine is still I mean, it's been changed somewhat, but it is still the vaccine which is our protection against inhalation anthrax, aside from antibiotics. So the second lesson is find a mentor. Now, in medical school, I met this gentleman, Robert Austrian, who came from Hopkins to Downstate Medical Center, Kings County Hospital, a hospital distinguished for many things, for example, having been built without surgical operating rooms, but that's another story. Anyway, Bob Austrian was a wonderful mentor, both in the laboratory but also on the wards. He was a diagnostician par excellence, and it was really a delight to see him do physical exams. But he also put bacterial polysaccharides on the map. That is to say, showing that invasive pneumococcal disease was still a problem despite antibiotics, and that a polysaccharide vaccine could protect, as he demonstrated in African gold miners. And the use of bacterial polysaccharides, as you know, began to generalize. After pneumococcal, it was Haemophilus influenza polysaccharide and meningococcal polysaccharide, both of which proved to be rather effective, in fact. But there was the problem of vaccinating infants, and the discovery by Oswald Avery back in the 1920s that conjugation of polysaccharides with proteins could improve the immunogenicity of polysaccharides was taken up by John Robbins and his associates, and so the first conjugate vaccines began to be produced. And as you all know, in the 1990s, these vaccines proved to be dramatically effective in controlling the 3 major pathogens, bacterial pathogens of childhood. My second mentor was this man, Hilary Kaprowski. Now, I told you that I fought to go to Philadelphia despite the advice of W.C. Fields, allegedly on his tombstone, rather here than in Philadelphia. But despite that advice, I fought to go there because I knew that Hilary Kaprowski had just moved to the Wistar. And Hilary was noted for his work, even then, on rabies and on polio, and I wanted to work with him. So I went into his office, basically, and asked him if I could. And he said, sure. So one experience that we had was to work on this virus, rabies. You can see the bullet-shaped particles of the rabies virus. At that point, in the early 70s, rabies vaccination consisted either of nerve tissue vaccine, 21 doses, or duck embryo vaccine, 21 doses, because they were poorly immunogenic. And of course, there were notable reactions, particularly to the nerve tissue vaccine, encephalitis, Guillain-Barré, and allergy to avian proteins were quite significant reactions. And I'll come back to this in a bit also, the rabies virus was adapted to growth in human diploid cell strains. Now, this picture shows Hillary and Tad Victor, who was a rabies virologist, and myself, and I was mock inoculating Hillary with the vaccine. In fact, we were the first three to take the vaccine, which was supposed to demonstrate our faith in the innocuity of the vaccine. This vaccine was essentially rabies virus grown in the human diploid cells and activated with beta-propiolactone, and it proved to be highly immunogenic. And this slide shows the antibody responses after intramuscular or intradermal inoculation, both of which are used today. The protective level is 0.5 international units, and you can see that a 4-dose series gives protective antibodies. And indeed, it so happens in a way by coincidence, that this 4-dose regimen is now in this country the standard regimen for protection against rabies. In order to collect these data, one of the things I did was to visit just about every veterinary school throughout the country, enrolling veterinary students in these studies. Now, I can tell you that veterinary schools are located in some out-of-the-way places, and it was rather, I would say, depressing to be in a place where the only thing open at night was the Dunkin' Donuts. But anyway, we got through those studies, and eventually the vaccine was licensed and has become the gold standard vaccine against rabies. Lesson 3 is be careful what you do when you're young, because it may come back to haunt you. Now, as I said, I convinced Hillary to let me into the laboratory, and the laboratory that I was mainly working in was the polio laboratory. At that time, Hillary was developing attenuated polio viruses for oral administration. And so in the lab, we learned a number of things: selection of plaque mutants, neurovirulence testing in monkeys, the schedule for immunization, including newborn immunization, which was a new thing at that time, the effect of maternal antibody on immunization, the instability of attenuation in the gut, that is, the selection of reverted mutants, the identification of those mutants, the fact that virus was excreted for long periods of time and could spread to contacts, mass vaccination, I'll come back to that, and the problem of interference with OPV in the tropics. Well, Hillary's competitor at the time was Albert Sabin. And as you all know, Albert strains did eventually prove to be the better strains, or, well, one can argue about that, but anyway, his were the strains that were adopted for worldwide use and have resulted in the control of polio throughout the world. So that was, of course, a kind of a competition similar to the Gallo-Montagnier competition of recent years, and it was certainly at times a rather heated competition. But in the course of that work, I went to the Belgian Congo to do the first mass vaccination, and the reason that we went there was because Belgian scientists had come to Hillary saying that there was endemic and epidemic polio in the Congo, and could something be done about it. So the mass vaccination was done there, and did show efficacy, although interestingly, we showed that the efficacy was only about 60% in that region, which is still a problem, that is vaccination in tropical regions. But it was a successful campaign. However, in 1999, a British journalist published a book in which he proposed the following hypothesis, that AIDS did start or was first noticed in the Belgian Congo, at least retrospectively, that the oral polio vaccine, the so-called chat strain, was studied in the Belgian Congo, that there was a chimpanzee camp associated with the polio work near what was then called Stanleyville, now Kisangani, and that the chimp simian immunodeficiency virus is genetically the closest virus related to HIV, so OPV must have been made in chimpanzee cells contaminated with SIV, which somehow then mutated to HIV. So that hypothesis received a lot of play in the newspapers and even in the scientific literature. So I spent about 6 months of my life, later in life, dealing with this controversy. And this is a picture of one of the items that I had retained in my files. I'm not going to read this, it's irrelevant. But the point is, I had fortunately kept a lot of the files that went back to the 1950s so that I was able to obtain information, retrospective information, which otherwise would have been lost. And that may be another lesson, never throw anything out. But the results of the investigations were that we determined, showed that no chimp cells had been in the production labs, that the chimps were used for neurovirulence testing, that PCR of the OPV lots used in the Congo were negative for chimp DNA, but positive, naturally, for macaque DNA, since the vaccine was made in rhesus monkey cells, that the same lots had been used in the US and Europe, without, of course, producing AIDS, that there, even in the Congo, there was no geographical correlation with vaccination. And working with Beatrice Hahn and others, molecular evolution studies suggested that, in fact, HIV started in about 1931. That is, crossed over from chimps in the Cameroon, west of the Congo, not the Congo itself, because those are the chimps that carry SIV most closely related to HIV, acquired because of the practice of butchering and eating chimpanzees in the Cameroon and indeed in the Congo. Lesson 4 is the heresy of yesterday is the gold standard of today. And that relates to something that was happening at Wistar when I went back there after doing pediatric residency and opened my own laboratory. And that was that Leonard Hayflick at Wistar was working on cell strains developed from human fetuses. And what he showed was that those cells had limited lifetimes. That is to say, they would replicate for a certain number of passages, usually about 50, but then they would die, just as we all do. And so that these were not continuous cells, but they were, so to speak, pure cells. At that time, the first simian viruses were being discovered, and the fact that primary cells could be contaminated with other agents was becoming an important problem in vaccine production. So I took one of those cell strains, called WI38, and applied them to rubella, because at that point in the early 60s, there was a huge rubella pandemic. As shown here, millions of cases, lots of complications in adults who had rubella, but also many infections in pregnant women, leading to thousands of fetuses dying in utero, and also at least 20,000 congenital rubella syndrome cases It says 5,000 therapeutic abortions, but I can tell you from my experience doing diagnostic virology during the epidemic in Philadelphia that there must have been many more therapeutic abortions than that throughout the United States. We're able to calculate that 1% of all of the pregnancies in Philadelphia at that point were affected by rubella infection. At any rate, so we adapted, well, this is a rubella cataract, cataracts in an infant born after congenital rubella. So working in the lab, we learned how to grow virus in large quantity, for example, in baby hamster kidney cells, the diagnosis of maternal infection, isolation of virus from the fetus, the importance of placental infection to fetal infection, the interesting fact that the virus actually was distributed through all of the organs of the fetus. Of course, with others, many others, we described the outcome of fetal infection and worked on the mechanism of persistent fetal infection. That is, the virus not only infects, but the cells remain alive and the virus continues to be produced. And what happens, though, is there is a mitotic inhibition and an apoptosis of fetal cells, which is probably the background mechanism for the pathogenesis of the various defects. And we adapted the virus to human cell culture, and passage of the virus showed that it, through various markers, that it was attenuated. And this became then a candidate vaccine virus. Well, of course, there were others working on vaccine development, and those strains were grown in primary cells. In 1968, there was a large meeting at NIH at which the various vaccines were presented, and Albert Sabin was there, and he was going through the halls and saying that we should not use human diploid cells, but, you know, what could I do about that? But then he, towards the end of the conference, he got up and he made this statement: there is no full characterization for any cell line, blah, blah, blah. There is always hypothetical something for which we cannot test. Therefore, the WI38 cells are no more fully characterized, blah, blah, and there's no technology, et cetera. Well, he did this in his usual rabbinical style, and but honestly, I remember very clearly, I said to myself, the Lord has delivered him into my hands. Because I got up and, among other things, said that despite my great and sincere respect for Dr. Sabin, I think the statements he has made are strictly ex cathedra and without a factual basis. So my conclusion is what we are dealing with here is theology. And you see in theology it is very difficult to disprove the existence of ghosts, but this is not, to my mind, a basis for making intellectual decisions. Now, this was certainly an impudent thing to say for a young man, but I received an ovation. Now, the ovation was not for me personally, but because most of the audience agreed with me. However, the FDA agreed with Sabin. And it was only something like 7 or 8 years later, after the vaccine had been used extensively in Europe, where they had a much broader point of view. It was only then, and because of the better immunological properties of the particular so-called RA273 strain, that the vaccine was adopted by Merck in the MMR vaccine. And the happy end of the story is, or at least towards the end, I hope, of the story, is that this is the map of countries now using rubella vaccine. And you can see that the only notable exceptions are sub-Saharan Africa and India, and that there is actually a reasonable argument for thinking that rubella could be eliminated from, and congenital rubella, more importantly, eliminated from the world by vaccination, particularly with together with measles. Lesson 5 is don't get into a mental rut, because having worked with diploid cells for so long, and then beginning to think of developing a varicella vaccine, we made a mistake. Now, we found, and this was the abstract that we published, we found that varicella virus could be grown in guinea pig cells. And that was sort of interesting, but we didn't think about that deeply enough. So at that point, Michiaki Takahashi in Japan was trying to develop a varicella vaccine. So was Maurice Hilleman at Merck, and also ourselves. And as you can see on this slide, both the Hilleman strain and the strain that we were working with, Webster, was serial passage in WI38. But Takahashi had grown it first in human cells, but then passage it in guinea pig cells. And he achieved what we did not. That is to say, the vaccine virus became attenuated, having been passaged in the guinea pig cells, whereas the two other strains were insufficiently attenuated, even after long passage in WI38. So again, don't fixate on things. Think about them before you try to repeat yourself. Lesson 6, I don't know whether it was Yogi Berra or Casey Stengel who said this originally, but it's not over till it's over. You all remember that in the late 90s there was a rotavirus vaccine called RotaShield, which was certainly effective. That is, it did show protection, but it also caused intussusception, and at a rate of approximately 1 in 10,000 first doses, and so it was taken off the market. It was actually offered to developing country manufacturers if they would use it on the grounds that it would prevent more deaths than it would cause, but in what might be called a profile in cowardice, none of them adopted or that offer from the original manufacturer. Well, so the need for rotavirus vaccine persisted. In the U.S., as you can see, the issue is hospitalizations rather than deaths, but many thousands of hospitalizations and medical visits, whereas the more serious problem in developing countries is something like 600,000 annual deaths and millions of medical visits, and of course, not always successful medical visits caused by the dehydration of rotavirus gastroenteritis. So, well, I'm going to skip that and talk about the development of the pentavalent vaccine. Working with two of my colleagues, Fred Clark and Paul Offit, at Wistar and Children's Hospital, and having as background the work of Ruth Bishop in Australia, isolating the human rotavirus, and the work of Al Kapicki in the NIH and his colleagues showing the importance of two surface proteins, the VP4P protein, the VP7G protein, carrying neutralizing epitopes, and having at our hands the technique of reassortment, we're able to make a vaccine consisting of 5 different strains. And that is shown on this slide. What was done essentially was to go to the veterinary facility of the University of Pennsylvania and to isolate from a cow a virus called WC3, which is shown in yellow. The rotavirus has 11 double-stranded RNA genome segments, and that makes it easy to reassort. So taking 4 human viruses, which have on their surface these G and P proteins, one is able in cell culture to make reassortments based on 10 segments of the bovine virus, which gives you the attenuation, and 1 segment from either a G or a P that is segments coding for either G or P proteins, so that one ended up with 5 different viruses, each one giving a particular immune response to the common human rotavirus strains. This was then tested in 70,000 infants to show that it did not cause a significant number of intussusception reactions, and the efficacy of the vaccine proved to be very high, as shown here, particularly 98% against severe disease, an effect lasting for at least 2 seasons. Now, the effect of this in the U.S. is shown on this slide, and actually the last season, 2009-2010, shows even lower rotavirus activity in the United States. And since not every child has been vaccinated, there is definitely a herd immunity effect occurring here. And of course, we are all concerned about serotype replacement, as has happened with pneumococcus, but so far that has not happened, and this success is now being repeated in other parts of the world, and especially now we have a second rotavirus vaccine that has also been proven to be effective. A problem has arisen similar to the poliovirus problem, in that the effectiveness of rotavirus vaccines in tropical countries is much diminished in comparison to its efficacy in developed countries. As you can see here in Nicaragua, the efficacy against severe diarrhea was 58%, against very severe diarrhea that would probably, without treatment, have resulted in death, 77%. The reasons for this are being studied. I'm trying to push Merck into doing a trial of neonatal doses of rotavirus vaccine, and we don't yet know what the biology of this is, but it will be very important to determine that. But nevertheless, if you look at the public health effect of even this kind of efficacy, the reduction of mortality is going to be considerable. Lesson 7 is be skeptical of expert opinion. Now, you all know that literally thousands of people are trying to develop a vaccine against HIV over the last 20 years or so. So what have we learned? Essentially what we've learned is that broadly neutralizing antibodies against HIV are difficult to produce. Nevertheless, in experimental animals, you can show that antibodies do protect. So that's important. The second is that cytotoxic T cells, if present at the time of exposure, decrease viral load after a challenge. But, not surprisingly, CTL does not prevent infection, but it does have an effect on the amount of virus. The other thing that we've learned from HIV vaccinology is the importance, the interest of what are called prime boost regimens. That is to say, where you prime with a vector carrying certain genes, the HIV genes, for example, or a DNA plasmid expressing HIV genes. And this priming produces at least a feeble, measurable immune response. But when you boost with another vector or with a protein, you get an enhanced immune response. And this sort of prime boost regimen, I think, has application to other vaccines as well. At any rate, in the 1990s, we were working at Sanofi Pasteur with a vector called canary pox, which, as the word implies, is a pox virus of canaries. And it has the property of being innocuous in humans, and also the property that vector immunity doesn't develop against it. That is to say, you can get repeated doses of canary pox and not develop a response that prevents the inserted gene in canary pox from being expressed and stimulating the immune system. So the Thai government and the U.S. Army decided that they would do a trial in Thailand in a population exposed to HIV using the poxvirus, canary pox prime, and the GP120 glycoprotein boost. Well, a who's who of HIV vaccinology, I'm sure you'll recognize some of the names on this list, published an article in Science saying this study should not be done. It's a waste of money, a waste of time, and it shouldn't be done. And you know, I respect every one of the individuals who authored this article. Nevertheless, at the end of the day, lo and behold, the trial showed an efficacy of somewhere around 30%. This is the first time that any efficacy has been shown against HIV acquisition by prior vaccination. And this despite, as at the bottom of the slide, you can see the CD8 T-cell response, which the experts say is absolutely essential, was poor. So why does it work? Why did it work? We don't know the full answer to this yet, but it appears that it works through stimulating antibodies and probably non-neutralizing antibodies, antibodies that act through antibody-dependent cellular cytotoxicity or perhaps other mechanisms. And interestingly, the antibody is directed not at the V3 loop, which is the loop that most people think is very important in inducing neutralizing antibodies, but the V2 loop, which is the loop that attaches to integrin. So in other words, it may be that this vaccine is preventing the attachment to integrin receptors on cells and therefore decreasing acquisition of HIV. But anyway, as I said, be skeptical of expert opinion. So my eighth lesson is be patient, follow your dream. In the 1970s, I started to work on cytomegalovirus. Why? Well, several reasons. One, to prevent congenital infection, which occurs in 1% of all live births and in many cases leaves sequelae behind, primarily mental retardation and deafness. Second, CMV is still the major infection complication of transplantation. Both solid organ transplants, recipients who receive an organ from a seropositive donor, or seropositive bone marrow transplant patients who are immunosuppressed and reactivate their latent CMV. And then finally, there are data suggesting that CMV may play a role in atherosclerosis and, important to some of us, immunosenescence. Starting in the 70s, we developed a live attenuated strain, and I won't go through these data, but the strain produced antibodies and cellular responses, and so that was the first step. We also used the canary pox vector to show that if we inserted the major stimulator of cellular cytotoxicity, the so-called PP65 matrix protein, we could induce CTL in volunteers, shown by the black triangles, after 2 doses of the canary pox vector. More important, at this stage, in 2010, the live attenuated strain is still being studied. It has showed protection against severe transplant disease, so that's one major finding. The third item, subunit GP glycoprotein, has shown protection about a short-term protection of about 50% in women exposed to CMV. So that's another proof of concept. And the next line, DNA plasmids in transplant patients, bone marrow stem cell transplant patients, has shown diminution of viral load and reduction of the need for antiviral therapy. So we now have at least 3 proofs of concept that a CMV vaccine is possible. And we have at least 4 major companies developing a CMV vaccine. So I feel in any case that I've been successful in getting things moving, and I hope to live long enough to see a licensed CMV vaccine. Lesson 9 is stay abreast of new techniques. As a consultant in this decade, I have had to deal with a number of new techniques, and I just want to mention 3. You all know that the papillomavirus vaccine has been developed using virus-like particle technology, basically a self-assembling protein, L1 in the case of the papillomavirus vaccine. But VLPs are being applied to many other viruses. For example, influenza, where 3 proteins are required, the hemagglutinin, the neuraminidase, and the matrix-1 protein, which can be assembled into virus-like particles and constructed in vitro. But this is also being applied to Ebola virus, to chikungunya virus, and even to HIV to make immunogenic particles that are not infectious. The second new technology I would mention is adjuvants. We have learned the importance of the innate immune system in stimulating the adaptive immune system. And so we are now dealing with adjuvants that are TLR stimulators, TLR agonists. They are mentioned at the bottom. We now have 1 licensed TLR agonist in the SmithKline papillomavirus vaccine, but also the TLR9 oligonucleotide, so-called CpG TLR9 agonist, is proving to be interesting, for example, in hepatitis B. This is a study in the elderly, and I must tell you the elderly are people over 40 who don't respond very well to hepatitis B vaccines, to the standard vaccines, but to a vaccine called Heplisav, which contains the TLR9 agonist, they do respond. So here you see that after a single dose of the TLR9 adjuvanted vaccine, 79% of the subjects respond with antibodies, and after the second dose, 100%, compared to only 18% with the standard vaccine. Now, this is going to be applied to diabetics, to patients on hemodialysis, etc. And this, again, is proof of the idea that the adjuvants of innate immunity are going to prove to be very important in vaccinology. And the third new technology is what might be called the omics. Reverse vaccinology, or genomics, was developed initially by Rino Rappuoli in Italy, and essentially what it consists of is a sequence of a bacterial pathogen, sequencing the genome, selecting the open reading frames that are likely to produce proteins, growing those open reading frames in E. coli, in other words, producing the proteins, injecting the proteins into mice, and then taking serum from mice and looking for bactericidal or other functional antibodies, and then selecting the proteins that give those responses and putting them into a vaccine. So Novartis now has a pentavalent meningococcus group B experimental vaccine, which is in phase 3. You know that we don't have a group B meningococcus vaccine. It's the missing link in our protection against meningococci. And this is, well, it says phase 2 in this trial, but it's actually in phase 3, and it appears to be doing quite well. But many other bacteria are also the subject of this genomic analysis: group B strep, pneumo, anthrax, chlamydia, and even Piriformonas gingivalis, the cause of a very important worldwide disease, periodontitis. And as I said, there are other omics, the transcriptomics, that is looking for messenger RNA expressed in infection, that is in vivo, so you know it's important, the structure of proteins, what are the important host responses to give you clues as to what the protective responses are, and even looking for the genetic, let's say the genetic structure of people who react to vaccines, who have adverse reactions. So, what are some of the future targets? Well, this is a mixture of, first of all, of vaccines that I think are likely to be licensed in the near future, dengue and meningo B, for example, but also future targets. For example, nosocomial bacteria, MRSA, Clostridium difficile, and we now have promising results in chronic infections. For example, women who are already infected and are showing neoplastic changes after papillomavirus infection. We know that the E6 and E7 genes of papillomaviruses are the ones that cause the oncogenic transformation, and there are already data showing that immunizing against those can actually, in a sense, reverse the changes that are produced by the viruses. And of course, many non-infectious diseases are the subject of vaccine development. So there are, however, remaining problems, and I've listed 6 of them that I think are important. The induction of long-term B-cell memory, both effector and central memory. The induction of cellular responses that are equivalent to those after natural immune responses, because when you get an infection, you mount a very significant response. We have not succeeded in duplicating that yet in our vaccine work. Overcoming poor neonatal antigen processing, because we do so poorly in the first year of life with immunization. Overcoming T-cell exhaustion in the elderly. Well, why is it that the elderly respond poorly to vaccines? It is, in fact, because their T-cells are naive T-cells are no longer being produced. Better induction of mucosal immunity with non-living organisms, and specific adjuvants that don't give you a broad response, but rather a targeted response to, let's say, dendritic cells, to T regulatory cells, Th17 cells, etc. Now, solving these problems is not necessarily going to be easy. Some of them will be very difficult, some of them will be intractable, but they will all be intellectually stimulating, which leads me to the last lesson, and that sometimes the reward is in the journey, not the arrival. And I think this is what Louis Pasteur meant when he said, young people, entrust yourselves to those sure and powerful methods of which we only know the first secrets. And whatever your career, don't let yourself fall into sterile skepticism. Don't let yourself be discouraged. Live in the serene peace of laboratories and libraries until the moment when you will have perhaps the immense happiness of thinking that you have contributed something to the progress and well-being of humanity. But whether or not your efforts are favored by life, when you approach the end, you must be able to say to yourself, I did what I could. Thank you.
Thank you, Stanley, for that wonderful tour de force. I think we have time for a few questions.
Left everybody speechless. Okay, well, first of all, let me say that we're still not out of the intussusception issue. Although we are now in a situation where if intussusception occurs, it occurs at a rate so low that it's going to be extremely difficult to determine whether it's true or not. we know certainly for sure that the two vaccines that we have, are not causing intussusception at a rate of, let's say, greater than 1 in 30,000 or so. As far as the efficacy is concerned, I do think that a neonatal dose suggests itself as one solution, because obviously there's going to be no interference at that point. And we know actually from Ruth Bishop's original work that that neonatal infection is usually benign. I mean, even with unattenuated rotavirus. So I think the safety is likely to be very high. So what are the other possible issues? Well, there's breast milk. That may be a problem, that is to say antibodies in breast milk. And there is a study being done of postponing breastfeeding at the time of vaccination to see whether that influences things. There's also the issue of maternal antibody. It may be that that women in developing countries have more passive antibody that they pass to their infants than women in developed countries. So that has to be sorted out. There is also the issue of OPV. Although it's been shown that there's no interference with the OPV, there is some reduction of the immune response to the rotavirus. Now, the trouble is that we don't have a good correlate for rotavirus vaccine protection. We have a surrogate, which is serum IgA antibody, and that is diminished. So I think there may be a problem with simultaneous oral polio vaccine. As we were discussing before, Matu, I think we should be using less OPV, so that may be one solution. But anyway, those are my suggestions.
That you would say, gee, if I had done it a different way, it would end up being better?
Oh, well. Yeah, one always has regrets. I told you about one with the varicella, where we should have used guinea pig cells. Okay, so with rubella, in fact, I know this may sound a little inflated, but the vaccine's actually better than I thought it would be. So I don't think I could have improved on that. With rotavirus, I think we probably started with the wrong idea, in the sense that I don't think we needed all of the P and G proteins that are in the vaccine, as shown by the competing vaccine, which, as you know, only has one P and one G. So I think it's probably a cellular response operating at the intestinal level that's important. And so I would have done that perhaps differently in retrospect. As far as CMV is concerned, at this stage, we don't know what's going to work, so it's hard to say.
Thank you so much for coming. It's been such an honor listening to you speak. I do have a question regarding the rotavirus vaccine. Is there any work being done? I know the RotaShield and RotaTech don't cover I think it's the G8 protein that's more prevalent right now in Kenya.
G9.
G9?
Yeah.
Okay. And I was wondering if there's any studies currently being done to cover that.
Well, you know, it turns out that both vaccines work pretty well against G9. So that's what I was just implying a moment ago, that it probably is not necessary to have specificity for the G protein. And so far, we don't have evidence that other serotypes, mainly serotypes that are prevalent in animals, are going to cause a problem. But as you said, the G9 is now very prevalent and is being prevented. The data are quite clear. G12 is also becoming important, and there aren't many data, but so far no indication that it's really going to be a problem. So all I can say is, at this moment, we're okay. What will happen down the line, we don't know for sure, but at this moment, I feel reassured.
I think at this point we're going to move on to our, Rebbe, you have one
question about the glycoprotein B vaccine.
Louder, please.
Question about the glycoprotein B vaccine for CMV. I'm wondering if there is any reconsideration about moving into phase 3 trial with the GB vaccine, given the new data on epithelial cell entry and its importance.
Okay, well, Ravit is sophisticated about CMV, and what she's referring to is that recently a new mechanism of entry of cytomegalovirus into cells has been demonstrated, and that is that not only is the glycoprotein important to attaching to certain types of cells, but also there is a protein complex, so-called UL-128-131, also involving a glycoprotein called GH, which is important in entering epithelial cells. Now, since the cells in which CMV become latent are mononuclear cells, mainly macrophages, it could be quite important to prevent entry of virus into those cells. And so Ravit is asking, is there reconsideration of using GB alone? And the answer is definitely yes, that at least 2 companies I know of are working on adding one of the proteins from that complex to a vaccine so that there would be an antibody response that would prevent entry into fibroblasts, but also an antibody response that would prevent entry into epithelial cells. So I didn't mean to imply that the CMV vaccine was solved, that we already have a vaccine, but only that we have proof of concept that a vaccine is feasible, because we have demonstrated protection with a variety of different approaches.
Thank you very much, Stan. I think at this point we're now going to call upon our panelists to come and join us. Stan has given us a really wonderful description of the last 50 years of vaccinology, and we'd like to talk a little bit about the future of vaccinology. So I'm calling up 3 faculty members, Dr. Ken Nelson from the Department of Epidemiology, and Drs. Anna Durbin and Oren Levine from the Department of International Health. And I think what we'll do is we're going to go in sequence, getting questions from Ken, and then from Anna, and then from Oren. I've told they're going to pepper Stan with questions. I've told him that turnabout is fair play, and he is, of course, welcome to ask questions back. After we've had a first round or so of questions from our panelists, we'll open discussion to the audience. So Ken, we'll start with you.
Okay, I wanted to start with a remembrance and a story. When I was living in Thailand, we saw a lot of rabies. In addition, my own pet dog developed rabies when we had scheduled a birthday party for my 8-year-old, and we had to deal with this. So we tested a couple of rabies vaccines, including the I think one of the first large trials of the WI-38 vaccine, and I was presenting these data at IDSA, and in the front row was Albert Sabin, and fortunately sitting next to him was Stan Plotkin. And Albert started the questioning by asking, how did you prepare this vaccine? Well, I didn't prepare it. It was Wyeth, but fortunately, Stan was able to help me out and make me look, make the talk look reasonable by some explanation of what this vaccine was. So that was one of my first encounters with Stan. But following up, I had a question, which I'm very interested currently in another virus that wasn't mentioned, namely hepatitis E. Hepatitis E virus turns out it's very important in Southeast Asia, developing countries, and now in refugee populations in Africa. It's very common. We also did a study in the NHANES sample and found 21% prevalence among Americans. So it's not unheard of here, although it's mostly asymptomatic. The problem with hepatitis E is that when it infects women, particularly late in pregnancy, causes a big problem. And there have been now 10 or 12 studies showing mortality of the women varying from 5 to 20%. So that's the problem. And the possible solution is that Glaxo has tested a vaccine in Nepal and found it to be 95% efficacious. But the market and what the Nepalese or the places where this is the problem populations would not support as a favorable economic venture. More recently, in the September, I think, 16th issue of The Lancet, there was a report from the Chinese Wantai manufacturers who did one of the largest vaccine trials it involved 111,000 people that were randomized to receive hepatitis B or hepatitis E vaccine. That vaccine was found 100% protective of those who received all 3 doses, and about 95% protective of those that received 2 doses. The problem is that it doesn't seem likely right away that people are moving, that this vaccine will become viable, or that other studies will be done. So my question is, what's the future of this, or how can we actually make this become reality?
So, Ken, the answer is actually implicit in what you were saying. Glaxo is not likely to undertake development unless their marketing department decides that there are enough people who can pay for the vaccine in the developed world to warrant the costs of developing the vaccine. They, as you said, they have shown that a vaccine can be made and that it's protective. So what's the answer? Well, one is if it could be shown that it is a significant problem in the U.S., if it could be shown, then marketing might change its mind. But failing that, it will be up to companies like the Chinese company to make a vaccine at a much lower cost and to use it in those areas of the world where the disease is important. Now, that means, and I'll be very blunt about this, it means that they will not have to satisfy the same regulatory requirements as GlaxoSmithKline. But that is the answer. Companies in the developing world to take up diseases that are important in the developing world and to make vaccines at low cost. The Chinese companies are beginning to do good development, but the problem, part of the problem, I mean, there are many problems, but part of the problem is that they don't do good clinical studies. And that's where they need improvement. And they need to publish in English language journals. Now, I don't say that out of any kind of chauvinism, but simply because People, by and large, don't read the Chinese language journals, and therefore the information such as you just conveyed doesn't get widely distributed. So this is part of the problem, but that's, I think, the ultimate solution.
Well, they actually published this one in the Lancet, so it was
Well,
well
yeah,
read.
but it's
Right.
not only a question of publishing the overall good result, but publishing the fine details
Right.
of what has been done.
Right. The other issue, the other impediment that I think even precedes the licensure of the vaccine is there's no licensed diagnostic test for hepatitis E, and yet it represents at least 25% of viral hepatitis is now classified as unknown. I suspect a lot of that is hepatitis E, but if clinicians can't test for it, except sending it to some research lab somewhere, that's an impediment. And I think that the importance of the disease will be more recognized by U.S. Public health and clinicians if there's a test. And it turns out in Europe they're using a test that's not licensed in the U.S. And in Germany last year there were 50 cases of hepatitis E reported. So I think there are a lot of impediments, and with Alan Labreque, we're hoping to move this, what I think is an important public health problem, forward. We have some data suggesting that 12% of maternal mortality in Bangladesh is accompanied by jaundice and is probably hepatitis E. So I think that's an important problem. But to move it has been difficult.
Anna.
Great. Well, again, thank you so much for coming to talk with us today. It really is an honor to have you here. You mentioned in your talk some of the advances and the upcoming advances with vaccinology, including adjuvants. But I wondered if you could talk a little bit about vaccine delivery mechanisms and sort of how that's become, I think, a very growing field, an important field in terms of vaccinology.
Yes, absolutely. We have reached the stage, certainly in this country, where I think we've reached the limitation of intramuscular or subcutaneous administration of vaccines, at least to infants. And so we have to solve or improve our delivery. So what are the possibilities? Well, the first, most obvious, is to make combination vaccines so that you're delivering multiple vaccines with one syringe. But that has actually proven to have some limitations too, because of interference between valences. So I think the main trends, in addition to combination vaccines, are first of all transcutaneous immunization, which looks actually quite interesting. As you know, Langerhans cells are great antigen-presenting cells, and so if you deliver vaccines either by patches with adjuvants or electroporation that gets the antigen through the superficial layers of the skin, or using microneedles to infiltrate the epidermis, then you get remarkably good immune responses. And there already is a licensed influenza vaccine in Europe based on a microneedle technology, which is going to simplify mass vaccination certainly against influenza. And there's no reason why it can't be applied to other vaccines. There's also aerosol delivery. Measles works well by aerosol. There are limited data, but I think very promising data with rubella by aerosol, and even non-living antigens by aerosol. So that's another train of development that's going on. Then, let's see, what else? Well, yes, of course. Now, adopting oral immunization is, of course, a very large area of exploration. And there, of course, one faces the dilemma that the gut has evolutionarily developed not to respond to foreign antigens, and yet you're trying to get that response. But there are ways around that, and this also gets into the area of giving vaccine antigens using transgenic plants as the vehicle, which is a field that still exists after many years, because clearly there are still problems of overcoming tolerance. But it's not without promise, especially in developing countries where one could feed materials that would contain vaccine antigens, and even though the response would be limited, if you give enough, then there could be sufficient responses. Plus, you have the prime boost effect. That is to say, if you gave an injection and then followed with oral material, you could get enhanced responses, and that could be another way of reducing parenteral injections, which, of course, also have logistic problems.
Yeah, yeah. Thank you.
Stan, thanks, and great to have you here. I think as I watched your life in vaccinology before us, I thought, you are one of the really unique people in the field who's been really successful in the academic field and then become essentially a captain of industry or a major force in the vaccine.
Lieutenant.
Not a kid.
In the industry, and I'm wondering with the young people in the room, et cetera, many of whom are probably thinking about careers in different industries, I wonder if you might just provide a couple of reflections or thoughts about what your major contributions were in academia and industry, and how, looking back, you think about the sort of contributions that people can make in either one of those.
Yeah, well, thank you. That's, of course, a very big question, and I'll try to give a relatively concise answer. There's an article written, at least the first author was Gordon Douglas, about the delicate network of vaccine development. And I think that delicate network expresses the concept. Basically, academia and industry do work hand in hand. I mean, that's what makes the vaccine enterprise successful. And so what does academia do, basically? The thing that the academicians do well is to discover the mechanisms, the pathogenesis of disease. That is, how does an organism cause the manifestations that are pathological, and what the mechanisms are that are involved, and what are the immune responses that protect somebody against a second exposure to that pathogen. Also, academia develops the data for the burden of disease, which is critical, as I referred to a moment ago. You have to have evidence that the disease is really worth preventing. And the example which is close to your heart is pneumococcal disease. I mean, once it was demonstrated that invasive pneumococcal disease was important, then many companies tried to develop vaccines against pneumococcus. Without that information, they would not have done so. So those are the important things that academia does. And so obviously, once having shown the immune response that is protective, then that gives you the indication of the antigen that you are interested in. And most academic labs will then go on to show in some animal model that whatever they're working with can possibly be protective. So at some point, industry becomes interested because they think that there's a sufficient market for a vaccine against a particular disease. Now, what I learned when I went to industry is that what I was doing in academia was pretty sloppy. That is that the kind of documentation, the kind of validation that one has to have to satisfy regulatory authorities is beyond what the usual academic lab is prepared to do. So one learns by experience in the industry how really careful one has to be. One has to have documentation of, what water you were using, where your reagents came from. It's really incredible how much must be done. But then industry has to repeat everything that's been done by the academic lab, essentially, to show that it really works, and then to develop a process of producing a vaccine on a scale that makes it feasible to license and to sell a vaccine. And that in itself is a difficult process, and there are a limited number of experts in that area of what's called development. And then, of course, you get to the standard phase 1, 2, 3, and 4 studies. But having said that, there again, the documentation, as academic investigators know, because they're usually the ones who conduct those studies, that kind of documentation becomes extremely developed and extremely large as well. And maybe the last point to make is that the whole process is extremely expensive. Now, this is not to make an apology for vaccine pricing, but the fact of the matter is that no vaccine in 2010 is going to be developed, well, let's say no vaccine that requires clinical trials is going to be developed in 2010 for less than $500 million. That's just the way it is. And some of them are going to cost more than that. So maybe, again, the last point is that companies have to make choices. And those choices are difficult if you're working in industry, because tomorrow you may be told to work on something else or that you were working on today, but that's just the way it is. So there are advantages to both, although I will say that for my own career, that going to industry when I was already, let's say, a mature investigator was a far better experience than going to industry when young, because you have more clout when you're older.
Thanks.
So I'd like to ask a follow-up question to that, Stan, which is just about the vaccine industry. I'm wondering if you could comment on what you think the health of it is right now, and in 10 or 20 years, what are we going to see? Are we going to see, are these various companies going to continue to merge? Are we going to see the emergence of companies in middle-income countries? What do you think?
Yeah. Well, I think the health of the industry is pretty good. And it's pretty good because of the prices that are now being charged for vaccines. I mean, let's face it, that's why is it that all of the major vaccine manufacturers are owned by pharmaceutical companies? It's because the pharmaceutical companies think that there's profit in vaccine manufacture. And in some cases, actually, the vaccine parts of the company are leading the pharmaceutical parts of the company. So I think the health of the industry is pretty good, much better than it was, let's say, 20 years ago. But for the future, there's no doubt in my mind that the future lies outside of the United States and Europe. It lies in countries like Brazil, India, maybe China. Um, Already, Korea has a significant vaccine industry because they can, at least at the present moment, make vaccines for less and therefore be interested in broader targets, and especially targets in developing countries that big companies won't touch. So I see the world changing in that respect in the relatively near future, where the world will become more dependent on manufacture outside of the U.S. and Europe. The problem now, but I think is in the process of changing, is that those companies outside of Europe and the U.S. are not doing a lot of research. They're mainly doing technology transfer. But that's got to change because the scientific abilities of people in those countries is clearly good, and so they will start doing research, and that will help because for hepatitis E, for example, I don't know what scientist Wantai has, but clearly they had scientists who were able to cultivate and inactivate the virus. Of course, hepatitis E, I might say, is a relatively easy problem. There are certainly much more complicated parasitic diseases and so forth.
Yeah, actually, the Chinese, it was a collaboration between a research unit at Xiamen University who actually developed the vaccine, and Wantai produced it and for the trial, because they needed, they needed 300,000 doses, so it was just to do this trial. But I had another question, and that is, one of the big issues is that a lot of preventable infectious diseases for vaccines that are available and vaccines that aren't yet licensed, like dengue, et cetera, occur in developing countries. And the issue of, the economic viability with the U.S. and European pharmaceutical firms developing, licensing, profiting, et cetera, supporting the research. Do you think the other partners like Gates Foundation and others they're playing some role now? Do you think that this will increase and be useful? Do you think this will overcome this problem?
Well, I'd hate to imagine the world without the Gates Foundation, first of all.
Right.
I mean, I think one cannot overestimate the importance that Gates has played recently in the world of vaccines, as you say, particularly in developing countries. The economics is not my forte, fortunately, but it is difficult to see how we're going to pay for vaccines for the entire world. I mean, if 6 billion people need vaccination, how are we going to pay for that? It's not immediately obvious. I mean, even Gates has some limitations on monies. So basically, governments are going to have to put a greater emphasis on health. I mean, they're going to have to accept the evidence that health is an economic benefit, and therefore prevention of disease is an economic benefit, and that they should be willing to pay for prevention. I see that as really the only possibility. We can't depend on donors forever. I know Warren probably has more of an insight into that than I do, but as I just said, I don't think we can depend on Gates. After Gates, what?
Right.
But there's one other point, perhaps, and that is that global warming is a good thing. because now we have dengue in Florida.
In Florida, yeah. Yeah.
And Texas, and so there will be a market for I'm
right.
being facetious, of course, but the fact that travelers travel to developing countries means that there is a market for many vaccines that there would not be a market for otherwise.
There was a hepatitis E outbreak on a cruise ship, and that helped from England.
Well, norovirus is an example of
Right.
a vaccine that I'm not sure just for you and me it would be a vaccine worth taking, but if we were going on a cruise ship or if we were going on extensive travel, I would certainly take a norovirus vaccine.
I think we have a question in the audience. Yes.
Just a quick question, separate from what you had said. The GSK malaria vaccine, RTSS, it's interesting with respect to the fact that it is GSK backed. It is a disease of developing countries. Granted, aside from reintroducing malaria into BC, it was endemic in the past. What is your opinion of the RTSS and its current efficacy?
Yeah.
As an executive advisor to Sanofi, would you have gone?
Okay, well, I gave you perhaps an overly cynical view because companies do embark on programs strictly for public health reasons. You could argue that no company should be spending money on HIV because, are we going to see a big market? Are people going to vaccinate themselves against HIV? Some groups will, but most of the problem is going to be in the developing world. So there is an aspect of public good in choices that companies make. And I have great respect for GSK and their malaria development. They've spent a lot of money over a lot of years in malaria vaccine development. And the RTSS vaccine, as you know, has shown moderate efficacy over a limited period of time. They are, again, as I'm sure you know, trying to improve with adjuvants the immune response, and that may help. But again, looking at it from the public health point of view, if you can decrease infection of infants over a period of time, perhaps using bed nets in addition, and then perhaps get into booster vaccination programs, you could have an effect on mortality and maybe even a herd immunity effect in terms of reducing the numbers of people who have Plasmodium falciparum in their bloodstream. You could have a major public health effect, even with a vaccine that isn't the best vaccine in the world. So I do see the RTSS vaccine as a major step forward. It's also obvious for an organism like malaria that one CSP protein is not the whole story. Right. So, whether it's AMA1 or one of the other antigens that would be added, I don't know. But eventually, I'm sure it's going to be a multiple component vaccine.
Yes.
Thank you very much. That was a very interesting talk. I saw early in your talk.
Is that Greg Glenn?
Yes.
Oh, hello. I didn't see you.
So I saw early in your talk you had mentioned maternal immunization as a strategy to protect neonates, and I know there's been some work done on that far long ago. And why isn't that blossomed more as a general?
It's because we have 500,000 lawyers too many. No, I'm actually serious about this. I'm not the only one. There are many others who have played more of a role, but I've tried at a certain point to interest companies, Sanofi and other companies, in maternal immunization, and the response is always the same. Well, our lawyers are afraid of the consequences. But I think there's glacial movement forward. That is, flu vaccine has been clearly recommended. Tetanus, of course, has been used for years. And I do think that there's hope for the future. That if a vaccine is given late enough in pregnancy, for example, an RSV vaccine late in pregnancy, or Group B strep, that there would be acceptance, and that perhaps the compensation system would put those vaccines into the system so that, unless the Supreme Court decides otherwise, that you couldn't go directly to a tort suit if your baby didn't turn out to have 150 IQ, because the vaccine hurt him during pregnancy. So it's a difficult problem because of the liability issue, but I hope it's going to be solved with time. At least I'm optimistic.
So maternal influenza immunization has created a very large safety database.
Yes,
Extremely
right.
valuable. It's also shown that maternal immunization ends up protecting infants from flu.
Right.
If you moved into RSV, you have a different paradigm. The benefit for the mother is not so well argued, and now your strategy is completely focused on infants. So that seems like a major paradigm shift and maybe more of a challenge.
You're absolutely.
Risk-benefit
Yeah.
ratio worth taking?
Well, yes, I think it is worth taking, but I agree with you. It does mean another step forward. But the problem is not the mothers, because the mothers, if you ask mothers, and of course this has been done, you ask mothers, would they accept that risk to protect their infants? Of course, they almost all say yes. It is essentially the liability issue if anything goes wrong at the end of pregnancy with the newborn.
Kate.
So I'd like to just actually extend on that a little bit. Dr. Plotkin, you've sort of pulled together a number of different observations and sort of projections for the future about the complexity of the vaccines that we're headed towards. Some of the pathogens are extremely difficult to understand how we're going to get vaccines for them. The increasing requirement for documentation and specificity in the regulatory pathway, the fact that where vaccine manufacturing is going to be headed in the future is away from the U.S. and European markets. And so I wonder, as I view all these things and as you've described them, we're headed to a quagmire where we demand more and more in places where we may not be able to do more and more. And I wonder, and for pathogens that are of increasing complexity, and I wonder whether you think that we're headed towards a necessary frame shift, that especially with respect to the regulatory environment, where I'm not sure that we have data that demonstrate that the increasing requirements are actually producing trials or products of quality that is unnecessary quality. We can always require more and more and more, and what is the metric that we're going to use to say enough is enough?
Well, Kate, your question, I'm not sure I can answer it because it touches on a societal dilemma, which we are in. And that is that the public seems to be demanding zero risk.
Right.
And there is no such thing. And the regulatory situation reflects that. And I don't have a simple answer to that. When people are queried about vaccine acceptance, et cetera, it turns out that, at least in this country, that at this time, in 2010, people are not focused on protection. They're focused on risk.
Yeah.
And that's the biggest issue that concerns them when a vaccine is recommended. So I'm really not sure how we get around that. As I said, the rotavirus vaccine required a study of 70,000.
Yeah.
New vaccines are probably going to require maybe not higher ante, but are also going to require large numbers of patients. and that's going to be expensive. So the answer may be, unfortunately, that clinical research and licensure are going to pass out of countries where risk is so much in the forefront. Although, as we were discussing before, those issues are passing to developing countries as well. So again, I just don't know how to solve that. We have to get across to the public the rarity of severe significant reactions, but also the inevitability of reactions in some people to anything that we give. How to do that, I really don't know. I will say that in December, I'm going to a meeting in London, which is supposed to talk about just that issue. How do you get this across to the public? I hope I come back from that meeting wiser than when I go in. Right now, maybe Neil has an answer, but right now I don't have an answer to that.
Let me
Yes.
ask a follow-up question. That is, I'm not so certain that the answer lies in your ability. I don't think the answer necessarily lies in your ability to communicate to the public, but I wonder if you could talk a bit more about the difficulties of the public understanding the science that's involved in assessing vaccines, and particularly vaccine safety, and the difficulty we face, and perhaps not have faced as squarely as we need to, of people in positions of respect at major universities who are doing what we would consider to be bad science and promoting bad science and getting that as accepted. What is our responsibility as a major academic institution here and professionals with dealing with these individuals? Because we haven't succeeded as well as we should.
Yeah, well, so, but, you know, doctors have traditionally been very poor at self-policing. I mean, you can see many examples of that. We just do a lousy job of controlling even just, frankly, medical practice. So, in a free society, I don't see how we can stop people from publishing junk, and especially, no, I mean especially since the publishing, the publishing industry is based on volume.
Right.
So, you have an incredible number of medical journals. When people say they can't get their work published, that's baloney. I mean, you can publish anything if you go to the bottom rung of journals, and I don't know how to stop that. I will say this though, and I'm sure you would agree with this, that it's the responsibility of the scientific and medical community to say very loudly when something is said that's clearly wrong. We're reluctant to do that. I mean, I know I'm reluctant to do that because, people will say I have a conflict of interest, this or that. But it really is important for people to get up and say, this is nonsense, this is unconfirmed science, this is false reasoning. And I don't think we do enough of that. My successor at Children's Hospital, whom you know well, Paul Offit, has become a spokesman.
Right.
Now, he does a good job, but he has been vilified, he's been threatened, and he has a lot of courage to continue speaking. Well, there should be more of people like that. I mean, there should be more people willing to get up and speak out. As far as making the public understand science, well, that gets you into the whole educational thing. We do a terrible job of teaching children about how to think about science, let alone scientific facts. So, I mean, We just have to deal with it in a more proactive way, and instead of just shaking our heads and saying, well, that's too bad.
I think we have to wind up. I thank you, Stan. I think your advice to speak out is a particularly wonderful piece of advice for all of our students here who are just starting their careers in vaccinology. We're having a poster session and reception in Finestone Hall. We'll see you all there. Thank you, panelists.